Welcome. My name is Francisca Acevedo, academic from Universidad de la Frontera, Chile, and I'd like to welcome everyone to the 6th BRT webinar on microencapsulation in pharmacy. As you may already know, this series of webinars have been organized by the Bioencapsulation Research Group Association and the Encap for Health Consortium. BRG has already organized more than 80 conferences, including training schools and industrial conventions. It has around 7,000 contacts from 80 different countries, covering around 60% industrials. It also has 2,200 members on microencapsulation LinkedIn group and also provides support to R&D collaborative projects. If you require more information on this, I invite you to visit our website, bioencapsulation.net. Encap for Health is an innovative sustainable encapsulation project for improving human health and well-being, which is funded by the EU's Horizon 2020 and is also present in 8 different countries, which brings together 12 expertises and around 30 researchers from different countries around the world. If you want to know more, please visit encapforhealth.eu. So, before we move on, I'd like to remind you of the rules for the webinars. First, remember that your microphone should be closed and that the chat may be used for technical problems only. Additionally, we will be taking all of your questions on the Q&A window. So, to begin with our webinar, I'd like to introduce you to Professor Felipe Oyarzun, academic from the Universidad de Chile and today's speaker. Professor Oyarzun has a PhD in research and development of medicines from Universidad Santiago Compostela, Spain. He is also founder and scientific chief of the Spinoff Austral Bioactive Materials and of entrepreneurship called Matetu. Dr. Oyarzun has vast experience on teaching sciences and pharmaceutical technology at Universidad de Chile and on working in micro and nanoencapsulation of biological active compounds. His main research lines are the encapsulation of active molecules in nano and microsystems mediated by non-covalent interactions, the study of chemical interactions for the formation of nano and microsystems, and the development of new materials for the pharmaceutical and food industry. Please welcome Dr. Oyarzun and his presentation, Micro and Nano Encapsulation of Biological Active Compounds, Motivation and Applications.
Hi, how are you? I'm Felipe. I'm a pharmacist, and now I'm talking from Chile. And it's gonna be a pleasure for me to present this work entitled Microencapsulation and Nanoencapsulation of Biological Active Compounds, Motivation and Applications. And thank you so much to the organizing committee for the invitation. Let's proceed. Okay, there is. Well, the research line that we explode in my laboratory include nanostructures done with different drugs, such as those hydrophilic, polyionic, and high molecular weight, such as DNA, RNA, heparin, those that are similar but not very ionic, such as proteins and peptides, other that have low molecular weights but that are also hydrophilic and ionic, but interestingly containing aromatic groups, okay, such as anti-inflammatories and therapeutic drugs, and also lipophilic drugs, especially those focused on cancer, such as docetaxel, paclitaxel, curcumin, among others. Well, we also explored the vehicularization of those nanostructures in microstructures or in different larger structures, such as microgels done by ionotropic gelation, and those such as powders in micro and nano size. Okay, in this case, the technology that we use is spray drying. Well, in the terms of this presentation, I will not talk about all of them and just consider these ones, okay? Well, firstly, let's go to talk about those molecules that are hydrophilic, not very ionic, and high molecular weights, such as proteins in the peptides, in the terms of multiple sclerosis, okay? And considering the drug, in this case, interferon beta. Well, as you can read here, this illness consists in that immune systems attacks the protective stealth, the myelin that lines nerves. It is represented here, okay? And this causes problems of communications between the central nervous systems and the rest of the body. Over the time, the disease can cause permanent nerve damage, okay? The signs and symptoms of this illness greatly vary depending on the magnitude of the nerve injury and which nerves are affected. Some people, for example, with severe multiple sclerosis may lose their ability to walk without help or to walk completely. Well, what is the situation and challenge? That interferon beta is the first line treatment to control this disease. But what is the problem or the challenge is it is very expensive, okay? And the administration is systemically, that obviously causing some adversities in the patients. Well, our proposal is the development of a nanoformulation for the direct delivery of the drug in the central nervous system by nasal administration, okay? And it will be tested in an animal model of multiple sclerosis, and we try to, once administered, it cross the bulb olfactory and goes to the brain. Okay, the development of the formulation consists in the taking advantage of the characteristic of the drug and controlling the acid-base behavior, controlling the pH, obviously, and promote their interaction with the negatively charged cyclodextrin to form a complex. And further, this complex between this drug and this drug in water will be able to interacting with chitosan, again in water, to form the nanoparticles. Well, here we can see some results about the developed nanoformulations containing another protein, a lysozyme, but also containing interferon beta from mouse or from human origins. And as you can see, the sizes are around 200 nanometers, showing low polydispersity and positive cytopotential. It's evidence that chitosan, that it's in the surface of the nanoparticles, and the association efficiency in every case is higher than 80%. Here you can see the images of those pictures by electron microscopy. Well, here let's talk about the therapeutic effectiveness of our nanoformulation. Okay. In this axis, you can see the clinical score, where 4 is very bad and 0 is a health animal. Okay, 4 means total immobility of the real legs of the animal, including the tail. 3, partial immobility of the real legs, including the tail. 2, total immobility just of the tail. 1, partial immobility of the tail, and 0, animal health. Okay. And as you can see here, when testing different formulations, the only one that could significantly decrease the clinical score is our formulation administered intranasally. In other cases, you can also see controls such as intraperitoneal administration of PBS, or intraperitoneal administration of the molecule alone, intranasal administration of the nanoparticles, or intranasal administration of just the drug, okay? And as I told you, our formulation was the only one able to significantly decrease the clinical score. Well, thus, as a conclusion of those results, our formulation induced a strong effect at significantly improving the symptoms of the animal, of the illness in the animal. Well, here you can also see that depending on the dose and the frequency, we can also control these parameters, even decreasing the dose administered previously. We can also improve significantly the effect, and it is important. Why? Because decreasing the administration from every day to every third day, and also decreasing the quantity of molecule, we are saving money and also improving the life quality of the patient. Okay. As I told you, we also can significantly decrease the schedule of dosification from every day to every 3 days, and a dose from this dose until this dose. It means about an 8% of the total decrease in the dose. Okay, let's talk a few moments about the conclusions and perspectives of the above development. It was funded by a project in Chile that is called Fondef-IDEA in the first stage. What we did, we request a PCT patent, an international patent, and we also published a paper, okay? If you want to look in deeper context, this you can see the reference here. And what is important that we test for this drug and this illness, but it also has the potential to be tested in another illness by the same way, such as cancer, Alzheimer, and Parkinson, okay? Well, let's talk now about the other kind of molecules that I told you. And those are represented by hydrophilic, ionic, aromatic, and low molecular weight drugs. Okay, what is important, these structures represent about 90% of the molecules that are available in the pharmacy to treat illness. Well, what is important also about this kind of molecules, as you can read here, considering the physicochemical characteristic of these molecules, their chemical entrapment in drug delivery systems by using, importantly, non-covalent interactions to provide control-release formulations represent an unsolved challenge for the scientific community. The above statement is based on the very low capacity of those drugs to undergo short-range interactions with excipients, okay? Consequently, occurs a very fast release of these drugs upon exposure to biological media. Very, very difficult to entrap these molecules in carriers, okay? Here I will show you some examples of these molecules: imipramine, amitriptyline, cyclobenzaprine, okay, antidepressive and muscle relaxant. And we are testing with 2 different kinds of polymers. This is PSS, you know, that shows an aromatic group. And this is absolutely the same polymer, but with the absence of this aromatic group. Okay, here we analyze by NMR how they interact if their interactions are intimate. This is the signal of the drug. This is the signal of the polymer. And this is the signal when we mix in aqueous solution the drug and the polymer. And as you can see, this signal is upfield shifted, what means that intimate interactions between the polymer and the drug is happening. Otherwise, when we're testing the same drug, but with the polymer without the aromatic group, the molecule is in the same place, thus indicating the absence of intimate interactions. What is important here that reflecting this result, we could mix the drug and the molecule, the polymer without the aromatic group, and there is absence of any interaction. While when we make this in water, we can obtain turbidity, and it is an expression of the presence of nanoparticles. Well, here is the same situation, but just exposing the turbidity that is achieved in the media. And here you can see the characteristics of the nanoparticles obtained with the 3 drugs, okay? Those are in the size range of 120 nanometers and 160, and with negative zeta potential, indicating that the polymer is in the surface. Well, here you can also appreciate that all the formulations are very stable, maintaining the size and zeta potential under different concentrations of salt, under different pHs, and also in different temperatures. This result is especially important because reflect that these formulations could be dried by lyophilization in the presence of a cryoprotectant, and then after adding water, you can obtain more or less the same size and zeta potential. And it is very important because indicate that it could be transformed to a powder and then, I don't know, could be sent to Africa, Australia, or United States in a cheaper way, okay? And it is more stable in addition. Well, here you can see some characteristics of our formulations. And as you can see, the association efficiency is very high. In every case, higher than 90%. The loading, it represents the weight of drug inside the nanoparticles, is also very high, very, very, very high. In fact, this value, it has been never evidenced before for low molecular weight molecules, such as the one that I told you. And this is an important, an additional parameter, and it is related of the nature of the drug interaction with the nanocarriers. Kinetic interactions means that the drug is physically interacting, and thermodynamical bound related to those drugs that the interactions with the vehicles is depend of equilibrium, okay? And as you can suppose, when the drug are physically interacting, trapped in the core of the nanoparticles, their release is very, very, very low. What is very important. Why? Because we are talking about drugs to treat chronic diseases. And if we are able to provide a very prolonged release of the drug, we can control the symptoms, and obviously it is better for the patient. Well, what is our proposal with this research? That depending if we create soluble complexes, nanoparticles, or massive aggregates, the drug release profile is gonna be different. For example, we propose that if you make complex, the release is gonna be achieved in days, in nanoparticles in some weeks or few months, and in massive aggregates, we propose that it could be from several months or inclusive. We propose 1 year, okay? And this just mixing in water the drug and the soluble complexes containing the aromatic group. Well, here we explore another important aspect of these molecules, and it is the capacity of those to be self-aggregated. Okay, those molecules exhibit this parameter, that is a critical aggregation concentration, and reflect how they have the capacity to form a polymer by themselves, depending on the concentration. This is the parameter that could be found for every molecule. As you can see, the tested molecule in this case are amitriptyline, sorry, promethazine, and chlorpheniramine. And as you can see, they have more or less the same size, the same polarizability, the same solubility, more or less the same quantity of hydrogen donor count or acceptor count, number of positive charge at the pH that we are working, that is physiological pH. But what is the difference? Look at this, that those molecules present more or less the same critical aggregation concentration. It means that at this concentration, they begin to self-aggregate. And these other have higher critical aggregation concentration. It means that it required higher concentration to be self-aggregated. As you can see here, testing the different molecules, you can see that at a lower charge ratio with amitriptyline and promethazine, the nanoparticles are obtained, okay? And those are that shows the lower critical aggregation concentration, while in the case of chlorpheniramine, that shows a higher critical aggregation concentration, the molecule, the nanoparticles are obtained at higher concentration of the drug and the polymer. And it is very important to elucidate this new critical role to creating more effective formulations, especially in terms of industrial capacities, because it provides a better performance and less requirement of drugs and polymers. Here you can see the characteristics of the obtained nanoparticles, and as you can see, the size is in the range of 120 and 180, and here is the number. Again, the polymer is in the surface, as you can see by the zeta potential, and this is the number of nanoparticles obtained. And as you can see, with those molecules, using less quantity of drugs and polymer, because they show lower critical aggregation concentration, the number of nanoparticles is more or less the same than here, thus reflecting the critical role to obtain more optimal nanoparticles. Well, what conclusions and perspectives about this finding? It was we obtained grants, funded, and also ring of research, okay? And the molecules that are available and that we are testing now are of a very large quantity, but among them atenolol, chloroquine, chlorpheniramine, amlodipine, doxorubicin, venlafaxine, and levodopa are very important because they work in very important diseases now, that are including cardiovascular diseases, allergies, cancer, Parkinson, and others, okay? Thus, the potential of these formulations is very, very, very high. We also apply for a provisional application, that is an international patent application, and we publish one paper. This is the reference if you want to check in a deeper way. We have a second paper now. We are answering to the reviewers. I hope to do that in this weekend, and a third one that will be submitted as up to a journal. Okay, and also we are analyzing the best strategy to develop implantable devices with a drug permeable pore. Why? Because as we are focusing in provide very, very long prolonged release, it's not possible to administering these molecules by oral way. Why? Because orally the drugs are eliminated in between 1 and 3 days. Okay, and if we are looking to control the plasmatic levels of the drug through time, it needs to be in an implant, for example. Okay, 1 minute again. Well, now let's talk about a different kind of molecules, and those are the lipophilic drugs, such as docetaxel, paclitaxel, and curcumin. Well, as you know, the main content, the main molecules that are contained in our body is water. Okay, thus, for a molecule to be able to generate an effect in the molecule, it should be dissolved or dispersed. Here we have a very old scheme about it, that here a molecule in an aqueous media must be solubilized or dispersed here to be able to interact with the receptor site to promote an effect. In other terms, what I try to say you that if the molecules are not solubilized or dispersed in water, it's impossible that they generate a biological effect. Okay, a biological effect mediated by a receptor. They obviously can trigger a physical effect, but not a biological one, such as the typical found with drugs, with common drugs. Okay, we proposed here by this way to create a system called nanoemulsions and nanocapsules. And the procedure is very simple and consists in adding an organic phase containing solvents, the oil and surfactants, also the molecule, in this case docetaxel, and this phase is transferred to an aqueous phase. Then, until evaporation, we obtain nanoemulsions. Okay, in this case, as we use a cationic tensoactive or surfactant, we can obtain a cationic nanoemulsion. As these nanoemulsions show cationic charge, a simple incubation with a solution that poses a negatively charged polymer, such as hyaluronic acid, will provide the effective interaction between this polymer with this negatively charged polymer and the cationic nanoemulsion to provide the nanocapsules. Okay, here you can see the different formulations that we developed, and all of them are in the range of 200 nanometers and 280, with very low polydispersity, negative zeta potential reflecting the location of the hyaluronic acid in the surface, and with association efficiencies in the range of 65%. Well, here you can see via electron microscopy that the systems have a round shape, and here you can also see the polymer that is in the surface of the systems. Here is this scheme of this formulation. Here is the drug located. Here you can see the surfactants, and here you can see the role of the polymer in the surface. And here you can see the importance of the filters for the obtained formulation. Among others, low polydispersity value, desired nanometric size, negative surface charge, and it is important because otherwise if the surface is positively charged, it is more toxic, okay? And it's also to very avidly interacting with the membranes, and we don't need that if we need to go into a tumor after intravascular administration. High encapsulation of the drug, enhanced uptake, and very good cell results, okay? Let's talk about those. Well, here we are testing the above formulation and loaded with docetaxel, that is an anti-cancer drug. Here in this axis, you can see the cell viability of a cancer cell, okay? What we want to do with these cancer cells to kill them, okay? And here's different doses of the different tested formulations. Well, as you can see here, here is in white the effect of the empty nanovehicle, no effect as you can wait. What is the problem? The problem is that when docetaxel is administered to the cells, there is no evidence of effect. Why? Because the molecule is lipophilic, it's not able to be dissolved or dispersed in the medium. Otherwise, when we put this molecule in this vehicle, you can see in black bars the effect of our formulation. This is a dose-dependent effect that is typically found for a formulation, for an active formulation, a formulation that is working in the cells, okay? This is a very important proof of principle. Well, now let's talk about another different molecule that is in some ways similar to docetaxel. Why? Because this molecule is also lipophilic. Well, curcumin, this is the structure, is an active molecule from turmeric. It is widely used as a condiment in various cultures. It is antioxidant, anti-inflammatory, and antineoplastic properties have been demonstrated for this molecule. Okay, what is the problem? That firstly, well, it is a very lipophilic, but also it is a very fragile molecule. It is very sensitive to light, to oxygens, to metals, and it is a problem, but it has a very high potential. It is interesting to note of this molecule that when it's consumed orally, its anti-cancer effect is especially identified at a gastrointestinal level. Why? This effect is topical in these cells because it is not able to be dissolved or dispersed. That is not possible to achieve this effect in distant tissues, just locally, because exists a direct exposition of the cells to the curcumin. Well, again, I'm showing that what happened when you expose this molecule to different solvents or a formulation. This is the molecule with water, and as you can see, almost all the quantity of curcumin is in the bottom of this flask, thus evidencing the low solubility. Here is the salt in DMSO. This is a solvent commonly used in the industrial pharmacy, but what is the problem? That is toxic. Okay, you can use that for make some controls or evaluating some mechanism, but it's not possible to create a formulation with this solvent. And here's how looks our nano formulation with the molecule inside. Okay, here you can see again similarly that I showed before that the molecule is dissolved in the oil nucleus, while this nucleus is separated from the aqueous medium by the surfactant. Okay, and here you can see how those formulation looks after examination by the electronic microscopy. Well, again, as I told you before, the elaboration procedure is very simple. In this case, it's even more simpler than before because it doesn't have a polymer in the surface. That consists just in adding this organic solution containing curcumin, the surfactant and oil and the solvents into water. Then after solvent extraction, we obtain the formulations. Well, again, as I told you before, it is possible to convert this aqueous suspension, it is this one, to a powder. And this is after freeze drying and in the presence of cryoprotectants. For example, here you can see the characteristics of the fresh formulation, and this fresh formulation is submitted to freeze drying and then redispersed in water. Again, the obtention of a powder is very, very important, especially for these sensitive molecules, because it is more stable in the powder, it's not able to be contaminated by, for example, bacteria, and it could be cheaply or could be transported in a cheaper way to distant countries, for example. Well, here we are testing in vitro results of this molecule, and as you can see, again, we are testing our formulation with cancer cell lines, and as you can see, our formulation kills the cells, the cancer cell lines, while not kills a cell line that not shows cancer cells. This is a cell line, a non-cancerous cell line. Thus, you can see that exists a different activity and is preferential active against bad cells, but not with good cells. Okay, and here we test our formulation, but without curcumin. Okay, this is the blank formulation in the different cell lines, and as you can see, the vehicle is very safe. Well, here we can see, using different technologies, that our formulation is able to fastly interacting with the membranes and maintaining the signal of curcumin in the membrane of cells. In very short times, maintaining the signal of curcumin, it means that it's stable, that it stabilizes the curcumin. Here is in the surface of the cells, analyzing by FACS. This is the name of the technique, flow cytometry. This is similarly to here, but inside the cell, okay, and comparing with the curcumin dissolved in DMSO, that is the solvent, the toxic solvent that I told you before. And as you can see also inside the cells, our formulation provides more stability to curcumin. And here we also demonstrate that after exposed the formulation to UV radiation by a laser, our formulation preserves the stability of curcumin inside the vehicle. And as I told you, as the molecule is very sensitive, the preservation of the activity also provides more potential to our formulation. Well, Here we also demonstrate that the formulation is also able to prevent the migration of the cells and also the invasion of the cells. Why is it important that curcumin inside our formulation could prevent migration and invasion? Because these are the key roles of cancer cells to produce the illness in the patients, okay? Because what happened that the primary tumor is able to move to distant tissues to create metastasis. And here we are preventing the mobilization and also the invasion of these cells in distant tissues that create that evidences more potential in our formulation to prevent or to treat cancer. Well, here we are testing in an animal model how our formulation could be able to prevent the recidivism and metastasis of cancer. This model consists in inoculate cancer cells in the animal, in this case the melanoma, that is a very, very aggressive cancer cell. What we do later, here the tumor is created, then at day 15, the tumor is extirpated. Okay, this is what a surgeon, what a clinician, a surgeon normally do when they identify tumor. What is the problem? That after this procedure in animals and in patients, obviously, after excising this and the saturation of the place, normally in a very large quantity of tumors, the tumor again is grow in the same place. That is very, very common, and this is called recidivism. And also after this new creation of the tumor in the place is also capable of creating metastasis in distant tissues. Okay, what again we will repeat this and I will show you the results. Normally we create a tumor, we extirpate the tumor, and at day 30-40 we analyze if the tumor there is a reappearance of the tumor in the place. And here you can see that every dot represents an animal. You can see that the 70% of the animals show reappearance of the tumor, and also the 70% of the animals show metastasis. This is lung metastasis, okay? Our proposal consists in just after excising the tumor and before suturate the wound, adding a very low volume of our formulation, 80 microliters, and evidence if our formulation is able to prevent the reappearance of the tumor and also is able to prevent the metastasis. As you can see here, our formulation completely prevent both important signals of the illness, okay, the tumor reappearance and the metastasis in the lung. Well, among the conclusions and perspectives of our formulation presented here, I could tell you that it involves two interesting contributions. Firstly, a technology to encapsulate and administering lipophilic molecules, and then that we also contribute with a proposal to applicate this formulation post-extirpation of the primary tumor. Well, among the results obtained for this proposal, we have one patent accepted and two patents application and several papers published. Well, from those patents, two of them has been licensed to the private sector, nanocapsules with hyaluronic acid to an European company, and the nanoemulsions with curcumin to a Chilean company. And in this case, we are able to create three products that are in the market containing curcumin. That is food with curcumin for animals and humans. And importantly, this food, as you could thinking, considering the above results, presents a biological and therapeutic effect, especially in terms of anti-inflammatory effect. Okay. Well, as you can see here, I will very appreciate the possibility to show, to present my results, to make this talk to you. And now I'm waiting for the questions that you can make to me. And this is my email if you want to write something, okay? Thank you so much.
Thank you. Thank you, Professor Oyarzun, for your interesting presentation. It's now time for the round of question and answer. So please refer to the Q&A window if you want to formulate any questions, and our speaker will answer them. So we have the first question. Okay, from Alan Connolly. Is it possible to modulate the release of the formulate nanoparticles? Professor Oyarzun.
Thank you so much. This question is very wide. What we try to do in our laboratory is the similar thing that a lot of technological laboratories makes, and is try to control the release. Okay, among the possibilities that exist, the one is, for example, to create. It depends tightly of the strength of interaction that is established between the drug or the active molecule and the excipient. For example, in the case of an emulsion, if the drug is inside the nucleus, you can prevent or prolong the release, coating the nanoemulsion with a polymer, for example, with a polymer such as hyaluronic acid, alginate, or carrageenan, chitosan, and others. In the case, for example, when the molecule is those very small, that very highly soluble, it is more difficult. And what we do to control the release and focusing on the development of an implant to obtain a drug release even for months, what we do is create not complex, but nanoparticles and larger particles, and we evidence that as the molecule is inside the disposal and is very tightly and physically trapped there inside, we can control in a very tightly way the release. It's gonna depend of the molecule. It's gonna depend about the structure, the size of the structure, the molecular weight of the structure, and obviously it's gonna depend about why you need to prolong the release, or which gonna be the use. Okay, if you wanna prolong for 1 day, for 1 week, for 1 month, for years, it's a very complicated equation, but it's possible to do. But to do, you need to really control the physicochemical behavior of the components and the drug. Okay.
Okay, thanks, Professor Oyarzun. We have other question from Denis Renard. Which biopolymers are the best suited for pharmaceutical applications?
It's gonna depend. Those questions are very wide again. It's gonna depend about what use you want. For example, if you want to obtain an implant, you must, and it depends if the implant you want to be that the implant is biodegradable or not. You want a disposal that's gonna be subcutaneously implanted or not. Okay. For example, and those are totally different that they're required for the gastrointestinal tract. For example, in the gastrointestinal tract, you can use carrageenan, okay? But carrageenan is not able to be implanted because it creates a lot of inflammation. Depends about the use, what part of the body. For another example is chitosan. For example, chitosan is widely used as in terms of a transmucosal administration. But there is some concerns if you want to use such as an implant. Okay, firstly, it depends about what use you want, then how it's gonna be administered, the way, and then you can select the polymer. Okay, it's very complicated. It's not possible to say, okay, those are the polymers that that you can use every time in pharmaceutical field. But for example, there are two that are very common, that are the PLGA, that is biodegradable, but it's able to entrap mainly lipophilic drugs, okay? And this is the acceptation of PLGA is very wide. It comes from a big company called Evonik, and another very acceptable polymer to be used for every way you want is hyaluronic acid, okay? Maybe those are the most common.
Thank you. Thank you.
You agree with me, Francisca? You can also comment about it.
No, your answer is perfect.
Okay.
Victoria Define says, thank you for your talk. It was very interesting. Did you check solvent in the nano capsules obtained by nano emulsion?
May you repeat again?
Did you check solvent in the nano capsules obtained by nano emulsion? What is the allowed amount? What is the value you obtained?
What is the name of the student?
No, it's Professor Victoria Define.
Professor, very good question. Very important question. very important question. Maybe one of the most important questions that I received. Okay, what do we do? Yeah, exactly. As you're thinking, this is a situation, a very important problem or situation that we must resolve. Okay. For example, in the case of food, when we develop foods, we can eliminate the acetone. As I show you, when we make the nanoemulsions and nanocapsules, we normally use acetone and ethanol, okay? But depending on the molecule, for example, testing the molecules that we are interested, we don't use acetone, okay? We just use ethanol that is not a large problem to be used in food. And in addition, after testing the quantity of ethanol in the concentrated nanoemulsion, the percentage is very low. I think that 5 parts per million. It's a very low content. Okay, but you can change the mixture of solvents. You can also use one of them and not the other, for example, acetone. But finally, the content is very low, but exists. Okay.
Okay. Oreste Piccolo asks, what do you think on the use of Alginate or maltodextrin instead of HA to encapsulate lipophilic drugs to have nanoemulsions of lipophilic drugs?
Well, thank you, Oreste, for the question. It's gonna depend. Okay, when we select a polymer or an excipient to be used in the vehicles, it's not just because we need a polymer. Normally, we select special characteristics of the polymer that helps to your medicine to go to a place to improve the effect. For example, in the case of hyaluronic acid, it avidly interacts with some receptors that are overexpressed in cancer cells. These cell receptors are CD44. And alginate is not able to interact avidly with those receptors, okay? It's possible to use, but it's possible to use alginate. It's cheaper, but we lost that special characteristic of hyaluronic acid. For example, I'm gonna give you another example. For example, in the case of chitosan. Chitosan is positively charged and not as alginate or hyaluronic acid that show negative charge. In the case of chitosan, it's more it provides mucoadhesiveness. Okay, it always depends about what are you looking for with your vehicle and with the polymer. Okay.
Okay, thanks. And the last question is from Marturo Shan. Thank you for your presentation. the ingredients used are registered for organic use?
Oh, I really don't understand the question. In what terms registered? I really don't understand the question. In fact, I do not know that exists a registration for organic use. Maybe someone could help me with that question. No, really, I don't understand what is the meaning of that. Sorry.
Okay, yes, I don't know.
Sorry for that.
Okay, I have a question for you, Professor Oyarzun. I'm very curious about your spin-off of Austral Bioactive Materials and Machetun. Could you talk about that a little, please?
Yeah, okay. Well, in the case of Adam, Austral Bioactive Materials, it is a spin-off that we create in the University Austral of Chile, in another university when I was a teacher there. It was the first spin-off created in that university, and we were focusing on help to the industry to resolve problems and to make solutions with a multidisciplinary group of academics. We were chemists, pharmacists, biologists. And so, for example, what we did, we create a patch that is able to be used for patients that suffering for a venous ulcer. That is a very large problem, okay? And it was a very, very effective. In fact, it helps a lot of patients to prevent and to cure that wounds, and it is already being used, and we try to transfer this patent to the private sector, but it's not easy to create that. Another development that we did is to create a box comprising alginate and salt that and this box is able to dissolve very, very, very slowly in the sand of the sea. And inside this box there are some seeds of mussels, and at the time that it was dissolved, the box, the mussels were growing. Thus, after, when the mussels are enough large, the tide was not able to bring the mussels to another place, okay, because they were culturing the mussels. In the case of Machetun, it is an enterprise, and I'm the scientific director. It is interesting to tell you that we obtain a result, the one of curcumin that we were talking about, and we had a lot of press, a lot of journalists make interviews for us, and by the radio was now the chief of the company. He heard me in the radio, then he saw me in the TV, and then he went to my office in the laboratory, knocked my door and said me, hey, I want to create an enterprise with you. Okay, exactly. Then he licensed our patent, and he was able to obtain money for other people. Okay, in fact, one of the investors is the man that create WOM, the company of mobile phones here. And then we create three additional products, and now we are exploring other areas. Okay.
Thanks. Very, very interesting. Thank you. And we have just time for the last question from Benjamin Moras. He says, hi, thank you very much for your presentation. What is the duration of stability of dried nano emulsions that have been freeze-dried? Okay,
Benjamin, years. This is the real stability of the dried particles. The lyophilization is a very, very, very useful methodology. When you dry your nanoparticles, and when you put in a bag without oxygen and light, it's gonna be preserved for a lot of time. Okay, and here I saw a complement of Marturo, and it was related with the use of the organic animal production. Yeah. Normally, all organic components that we buy are able to be used for animal production. Yeah. The question the answer for the first question is yes.
Okay. Thank you, Professor Oyarzun. Thank you for the questions and participation. And before we finish, I would like to remind you of the upcoming events. Our 23rd Microencapsulation Industrial Convention will be held online in June 2021. It's very important your participation in this convention, particularly for industrials. Our 24th Microencapsulation Industrial Convention will take place in the Netherlands from May the 2nd to May the 5th, 2022. And our 27th International Conference on Bioencapsulation will be held in Bielefeld, Germany in spring 2022. The next webinar on innovative particle capsule engineering using membrane emulsification will be held on May the 12th, 2021 at 3 p.m. Central European Time. Should you require more information, please visit our website bioencapsulation.net for registration and suggestions of more meetings and webinars. So that will be all for this BRG webinar on microencapsulation in pharmacy. Thank you, Professor Oyarzun. Thank you all so much for attending. Bye-bye.
Thank you so much.